I think we'll go ahead and get started. It's the top of the hour. I'm really delighted to have Dr. Tom Sellers here. He received his PhD from Tulane University in the area of epidemiology, and then he went on and did a postdoc at, let me see, my glasses aren't really that great, at Louisiana State University in the area of genetic epidemiology. He currently is the Center Director and the Executive Vice President of Moffitt Cancer Center. And he has academic appointments as professor in the department, or actually in the College of Medicine and also the College of Public Health at the University of South Florida. He has really strong ties to Minnesota, and that's one of the reasons why I'm so delighted that he's here. He actually was the program leader for prevention and etiology when the Cancer Center first started as a designated NCI Cancer Center. And he also then became the Associate Director of Population Sciences here at the University of Minnesota. And in fact, he was one of the members that had submitted the first CCSG proposal to NCI, and we had, or the Cancer Center was lucky enough to receive that grant on the first try. He then went to Mayo, and he was the Associate Director there at Mayo Cancer Center, and then subsequently became the Deputy Director. And then he landed at Moffitt. Now, Dr. Sellers is on numerous external advisory board, and in fact, he's on our external advisory board at the Masonic Cancer Center, too, so we're really delighted to have him as one of the members. Dr. Sellers' primary area of interest is cervical cancer, but he certainly has done research in other cancers such as breast, lung, and prostate. And his research, I think, is really a clear example of team science. He's really interested in looking at some of the inter-individual differences in susceptibility to cancer by examining genetics, nutrition, alcohol, smoking, and other risk behaviors. And he hopes that this type of research will inform how we actually approach cancer prevention, as well as improving cancer detection and improving treatment outcomes through personalized medicine. So I think this talk that he's going to be giving today is going to be a real clear example of multidisciplinary approach to science. So we're really delighted to have him here. So please welcome Dr. Sellers.
Dorothy, thank you for that very kind introduction. I was reflecting on the fact that I think it was 30 years ago, almost to the day, that I was coming up here for interviews. And I was a postdoc in New Orleans, a little bit concerned about the weather. And so I called ahead to the recruiter from the Division of Epidemiology, and she says, "Oh, Tom, it's great. It's going to be 35 degrees. And I said, "Oh, shirt sleeve weather." And she said, "Well, actually, you will see people in short pants." And I did. And it was snowing. And I thought, oh, this is going to be tough. They didn't tell me exactly how to adjust, but I made it 9 winters here. And I was kind of proud of that. For a California boy where snow is something you drive to, it was a bit of an acclimation. But wonderful time here, and it's really great to be back. Okay, let's get started here. I am presuming that this is not a room full of epidemiologists. I'm going to assume that I know there are a few people who worry about ovarian cancer, but the majority do not. And so I'm going to just cover a broad swath at about a 30,000-foot view, and hopefully I will have insulted almost everybody once. That's kind of my goal, say something irreverent, and I really don't want to do that. Okay. First, the epidemiology. This shows the incidence of epithelial ovarian cancer, statistics from the American Cancer Society. You see over 22,000 new cases a year. The incidence is actually decreasing. So when you're writing your grant applications, you usually have to say, this is a really big issue, right? And ovarian cancer is not in the top 10. It's like, uh-oh, that's not good. That's not going to convince them. But when you look at mortality, it is. It shows up as number 5, fifth leading cause of death and 5% of cancer deaths. So that is not a good thing and a reason why we need to understand it a little bit better. So we know that genetics play an important role in ovarian cancer etiology, and we know that because of the identification of high-risk, high-penetrance gene mutations like BRCA1 and BRCA2, and the lifetime risks are elevated quite substantially. You see almost 40% for BRCA1 and 11 to 17% for BRCA2. But there are other gene mutations that have been identified primarily through family studies that have significant risk. They're related in the DNA repair kinds of pathway if you tested candidate gene hypotheses, and you see the lifetime risk is 3 to 10%. So also very significant. But there are gaps, right? We're all here because we're in the business of science, and so we have to find out what the gaps and keep moving the ball forward. So in terms of major genes, in terms of multi-generation pedigrees, they have been tortured. They have not confessed. It does not appear that there are likely to be any other further major penetrance genes that are at least specific to ovarian cancer or breast ovarian cancer. You could then say, well, what about other genes that are in the DNA repair pathway or something like that, modest risk genes with lower penetrance? And it is possible. They don't seem to be coming in at a high rate of return, but it's still out there. And the reality is when you look at twins and try to estimate the proportion of the disease that is heritable, it's estimated that it's 30%. So clearly there are some gaps out there that have to be identified. And the question has become what and where is the missing heritability? So that takes us into the dark matter. And you know, there was a lot of bad press when we realized after mapping the human genome that only about 1 to 2% codes for genes. And so the rest, well, we didn't know what it meant. It must be junk. And people were publishing books, and I don't know if they're making a lot of money, but they were getting on TV talking about how bad we were and how we were totally missing the boat. Turns out the dark matter is quite active. And thanks in part to the ENCODE, we have indices of about 80% of the genome that is actively transcribed, or at least shows evidence of biochemical activity. So it's clearly not junk. It's just playing a role that we do not understand. I see a couple heads nodding, so we're with me so far? Nobody's offended yet? Okay, good. All right. I'm going to keep going while I'm hot here. So we came to that conclusion through an epidemiologic approach using genome-wide association studies. So who here does GWAS? A couple people? Okay, so you guys can check out for a couple minutes while I explain to this on one slide what it is that we do. So this is the recipe if you want to do one of these at home. Just follow this. It's really simple. You need a large number of cases and controls, and this is one of those times where it cannot be too big a study. Okay? You will not get penalized by the IRB or the SRC or your biostatistical colleagues. Don't worry about it. Then what you want to do is interrogate the genome for single nucleotide polymorphisms, and that is shown here where there's a single base pair change between individuals in the population. Not that it is the cause of any variation or risk, but that it is a signal, a signpost along the 3 billion base pairs that we have in our DNA. About every 1,000 base pairs, if we can characterize one of these SNPs, we'll measure it. Typically on something like this. This is one of these gene chips that you can now tag 500,000 SNPs for about $30. It's really a great bargain compared to the olden days when I first started. And then what you do is statistical analysis to compare the allele frequencies between women with the disease and without the disease. And if the frequency of this variant is higher in the cases, we conclude that there is that SNP or one nearby is increasing risk. If it's less common, then you would say that is protective. It's inversely associated with risk. So you're doing 500,000 statistical tests. How do you look at your results? Not a page at a time or a SNP at a time, right? You do it graphically. You look at pictures. So this is a picture of data from a GWAS. This is actually from the first ovarian GWAS that we did, where along whoops, go back. There. These are chromosomes, and then these are p-values, and each dot represents a single SNP. And so what you're looking for is a spike like this, where it is beyond 10 to the minus 8 p-value. And that's because you're doing a lot of statistical tests, a lot of false positives. How do you get the large sample sizes? You have to collaborate. That's team science. I've been fortunate to be involved with the Ovarian Cancer Association study consortium, which began in 2005 for those of us who were chasing candidate genes. And we realized we thought 1,000 cases and 1,000 controls, that's huge. and it wasn't, and we were torturing data. We were not finding anything, so we got together and said, well, why don't we compare our hypotheses? Why don't we compare data? And we started doing that. Then we started doing genotyping in our individual labs, sharing it, and it got to the point by today, actually about the last 6 years, we would send our DNA to a central laboratory, and they would do all the genotyping, and then we would all chip in on the analysis. Okay, here's the results. 10 years on one slide. I'll give you a second to read all that. Now, look, I know if you're in the field, you know what all of this is. If you're not in the field, you're going to go, so what? There's a lot of data there. So let me try to illustrate a few of the really key points here, I think. First of all, the first locus that was identified was on chromosome 9p22. The gene, at least the nearest gene, is BNC2, so it's basonuclein 2. So in the era of candidate genes where we think we know something about the biology, does anybody know what BNC2 does? You can't answer. It's involved in freckling. Yeah, so that's, of course, that's related to ovarian cancer risk, right? No, nobody, I mean, if anybody said they would have had an a priori hypothesis, I'd say, you're lying. It's not true. But it proved that this worked, right? Okay. Now, we have surgeons in the audience and clinicians who know that epithelial ovarian cancer, I said it's a single disease, but it's clearly not a single disease. And a pathologist can look at this under the microscope and say, oh, yeah, these are all different histologies. There's now emerging evidence that there are different tissues of origin and from different parts of the ovary. So there's a lot of emerging evidence that this is true. And it turns out that is emerging from the genetics as well. So we did post hoc analysis of the first stage 1 GWAS and identified that there were some loci that were specific to a certain histology and completely unrelated to others. So that was an interesting observation that led to the way we did our future studies. The MYC region is a real interesting locus on 8q24, and that's because it is associated with a number of different cancers, right? But it's not the same locus. It's near 8q24, but it may be on different sides of the MYC locus. And so it seems like, hey, there are specific regulators, perhaps, of the MYC locus that is raising these cancer-specific associations. And then just a little plug for epidemiology. So in the field of GWAS, there were really two camps, people who come at it from a purely genetic standpoint. And the thought was sample size trumps noise, right? There's all kinds of study designs. It didn't really matter. The patients could be recruited from, didn't matter, samples fell off the back of a truck. We'll find the signal if it's there. And that has proven to be true. When we started our GWAS, though, we were very explicit about inclusion-exclusion criteria, incident cases, not prevalent, certain histologies, try to rule out BRCA mutation carriers. And our top hit was in something called TPARP at that locus. We were collaborating with our colleagues at the UK, frenemies. No, they're wonderful colleagues. In their first phase, it came up as the 42nd thousand in the ranking, right, of the hits. That was overall, and then among serous, 182,000. So it didn't bubble up to the top in any stretch of the imagination. But when we pooled it, it was significant. So just as an aside, TPARP was identified when these colleagues exposed tissues to dioxin, right, a known suspected carcinogen. And it bubbled up as the first protein that was expressed when you expose these cells with dioxin. So we thought, well, isn't this interesting? Maybe there is genetic predisposition to certain exposures, and genetics can unmask it. We haven't been able to prove it, but we thought this is really cool. So if somebody's inspired, please follow up on this. We have some people who worry about chemical carcinogenesis in the room, I think, right? Steve, you want to chase this down too now for us? No, okay, I'll work on you later. Okay, so we got interested in microRNAs, in part because Craig Mello gave a talk at Moffitt, and this guy won the Nobel Prize. He shared the Nobel Prize with Andrew Fine for identifying these. And we thought, we're talking about the non-coding genome. We wonder if microRNAs play a role. So microRNAs, for those of you who are not familiar with it, relate the transcription, right, of signal. So they're encoded in the nucleus. They're transported into the cytoplasm where they're modified, and then they get out here. And if they bind to the seed regions of target genes, and they bind perfectly, it blocks transcription. If there's some variation in that seed region, they partially bind, and they have an effect on the end product, the quantity. So we thought, well, that's really interesting. So we did, there was this large collaborative chip called ICOGS. We had some real estate on there where we put polymorphisms in seed regions of genes, microRNAs that were relevant to ovarian cancer. And we found something, right? So this is what we found. There's this large inverted region that's common to Europeans of 22% allele frequency. So very, very common. There's been no crossover events in millions of years. And the arrows are pointing to, there's actually 4 dots here that were genome-wide significant associated with ovarian cancer. And we got really excited. I mentioned that this was a collaboration. The ICOGS was a number of different cancer sites going together. And the coordinating center was at Cambridge. And as we're working on the paper, they said, well, you can't really write this up as an independent locus. We said, well, why not? They said, because there's other SNPs that are nearby that are more strongly associated. Well, we didn't know it because it was nominated by another group. So this is the other group. So it was the breast cancer group. They were chasing something down. And the coordinating center said, you got to put this in light of the other day. Well, we didn't know it existed. So statistically, they are so closely associated on the same chromosome that if you measure any of these, you can't tell where the signal is coming from. So we published our findings, and we don't know if it's independent or not. So that's another thing that I'm hoping somebody follows up on, okay? It could be a different locus. We just haven't been able to prove it. This is good colleague and friend, late Kathy Phelan, who died shortly after she led this paper, which reported findings of 12 new ovarian susceptibility loci. So this is what you need to remember, take-home points, okay? There are 27 robust SNPs that have been identified today. There's a couple that are in press that we think will add to that number. The associations do vary by the histotype of the tumor, and it's sometimes in the opposite direction, which is really very interesting about what does it tell us about the biology. I didn't show you, but we've done an analysis that compared loci that were shared with prostate cancer and breast cancer. And so there are some regions that it's the same locus. So that's called a pleiotropic effect. We need to do that for other cancer types as well, right? I mentioned freckling. So maybe melanoma, there may be something shared with melanoma, who knows? So the magnitude of risk, and this is important clinically, the magnitude of the SNP effects that we're finding are similar among BRCA mutation carriers and the general population. So that does help predict the variability in lifetime risk. We've got pedigrees with high risk, and the age at onset distribution is all over the place. And it may be that these other SNPs are contributing to that. At the end of the day, where do we stand in terms of the 30% heritability? We've got about 6.4%. So we've got some work to do, right? So how are we going to find them? So this is where, in California, we would call it free thinking. We've got to come up with something different. But this is integrative molecular epidemiology, because when I told you, if we're measuring 500,000 SNPs at once, and we're now imputing to nearly 20 million on an individual, what percent are going to be significant at the 0.05 level? 5% of 500,000 is, where's the biostatisticians? They do this in their head. Like 25,000 SNPs, right? And so you go further, there's a lot of false positives. So that's what's under the surface here. Say 10 to the minus 5 to 10 to the minus 8. They may be real, but we can't tell if it's real because there's so many tests being done that we're protecting against false positive discoveries. Come back here. So we need to think creatively about how we're going to identify those. We believe that one strategy to do that is to think more about the biology, right? Enhance the probability that it's not a false positive. There's some underlying reason why it could be true. This is a paper that was published in Cancer Cell a few years ago by some really very, very smart people on the role of long non-coding RNA. And we got intrigued by that because of one of the things here they talk about, the tissue specificity. And so we thought, why would we worry about a SNP affecting ovarian cancer risk if it wasn't expressed in ovarian tissue? And it could. I mean, you could spin out a yarn, well, if it's involved in immune infiltration or something, it could be. But why not start there? And so we did that. And this is where I get to talk about Brett Reed, who's a master's prepared data analyst who's now working on her PhD in epidemiology. And she led this paper a few years ago. This is the story. So we've got risk loci that we've identified across the genome. We can look at chromosomes, and we know the coordinates. We know where they exist. There's another database that you can go and identify all the long non-coding RNAs, and you know the coordinates of where they exist on the genome. And so you can ask the simple question. If this is a risk locus, are there long non-coding RNAs nearby within, say, a 500,000 base pair region in either direction, so that it may be by just greater than chance you would see these at the same locus? Okay, you with me? It's an ecologic correlation study. That's all. So if there's smoke, there's fire. So we wanted to ask, are there more long non-coding RNAs encoded where we have these risk loci? And right here these light blue bars you see for all invasive and for serous. That asterisk means it's statistically significant. Do the happy dance. That's good. But as I mentioned, not all lincRNA are expressed in ovarian tissue. So you can actually use a different database to find out is it expressed in the tumor tissue, in the normal tissue, and you see that the level of significance goes way up. So that led us to believe that we might be onto something here, and it's worth focusing on long non-coding RNA to help filter which of these SNPs that are less than 10 to the minus 8, higher than 10 to the minus 5 may be relevant. So how do we show some evidence for that? Come on, work with me here. Go forward. There we go. So one of the SNPs that emerged was in HOX-A11, the antisense. So this is the RSID. We found it in the US GWAS alone. The odds ratio is less than 1. That means it is protective. Yeah, inversely associated with risk. Didn't hit the magic 0.05 p-value. My crew would be way different if 0.06 was a convention rather than 0.05. Way different. I'm king of the 0.06. But the false discovery rate says even after all of the tests, this is probably believable. So if you look at normal ovary, you can see that it is expressed. But in ovarian tissue, you can see a little bit of expression here, but it's significantly downregulated. And if you don't trust your eyes, you can actually look at the expression here, and you can see that there's a lot of zeros before the 5, meaning that there's significantly lower expression in the tumor tissue, right? Okay, so that's good. So this is Ted Richards, who was a graduate student at the time. I think he's still working on his postdoc at MIT. So he took particular cell lines and transfected this SNP into these cell lines and injected them into mice. And what you can see is that with the common allele, it was smaller, and for the minor allele, which was associated with lower risk, even smaller still compared to the normal vector. And again, if you don't trust your eyes, you can actually weigh it. You can look at the size, and you can see that these are all significantly lower for the minor allele. Again, inversely associated with risk, smaller tumors. Okay, that makes sense. You can look at an invasion assay, and you can see that in the tissues of these mice that had the minor allele, you can see that there's far fewer cancer cells there. And you can also do an apoptosis assay, and you can see that there's far greater programmed cell death in the tumors where there's been transfected the minor allele. So again, all of this is consistent with this being protective against ovarian cancer. That led us to the current R01 that we are doing on epidemiology and biology of lincRNA. And so the first, and this is multidisciplinary, and that'll become clear in a minute. So what we're doing is taking all of the available GWAS data sets, which have increased in number since that first publication, and the finding of the number of long non-coding RNA continues to increase. So we're going to redo that analysis to find out which are the regions of interest. We also, the hypothesis here is that these SNPs, or whatever the causative SNP is affecting transcription of something that's increasing or decreasing risk. So how do you do that? Well, you need to look at this transcriptomic difference between tumor and normal. TCGA is a great resource, right? Cancer Genome Atlas. Anybody know how many normal tissues there are for those who worry about ovarian cancer? Anyone? Anyone? You can speak up, Heather. Yeah. It's 9. It's 9. There's like 450 tumors and 9 normal. Guess what? It might be important to have normal. So we're not going to get it from TCGA. So we're doing some transcriptomic profiling ourselves. And then what we want to do is look at these risk SNPs. Are they leading to changes in expression of target genes nearby at that locus? Okay? So it's easy when it's on the same chromosome. It's a little bit harder when it's on a different chromosome, although that happens. And then we do all kinds of functional analysis, allele-specific, like what I just showed you, the work that Ted Richards had done. And then we're actually making mice. It was really intriguing when the scientific review committee said, yeah, you're the PI of this protocol, so you need to show that you're proficient in handling mice. I'm not doing the mouse race. No, dude, you gotta know what they're doing in the lab, right? I don't have time to talk about this, but one of the things that's become really important is epigenetic silencing in the tissues in these regions. And so we thought it was complex, but it's now even more complex. And you're going to have to integrate epigenomics with germline variation in normal tissues as well as in tumor tissues. But we got to figure this out, right? This is important stuff. I said it was multidisciplinary. It really is. So these are my colleagues. I've highlighted Simon Gaither. So he's a molecular geneticist. He's out at Cedars-Sinai. He's got a strong lab. His wife, Kate Lorson, is also doing the molecular biology work. But we've got a mouse modeler, bioinformatics, biostatistics. You can't have too many people in that club. So it's really fun. I don't have any results for that yet, because we're in the 3rd year and we're still torturing the data and generating the data. But I've got other things I can talk about. In fact, they gave me 45 minutes. So one of the things that I wanted to talk about was just the whole approach of how we're tackling big data. So here are some of these massive data sets that are out there. Who's worked with ENCODE or used ENCODE for anything? At least 1, 2, 3 people, 4. Good. That's all? It's free data. This is great. Anything free, you just can't get enough of. So I would encourage you to, same thing with, I mentioned the Cancer Genome Atlas. Great source of information. And when we're doing epidemiologic studies, we're supplementing it by looking at these great resources. GTEx for genotype expression in cadavers. So that's been really interesting to get at what's going on in the normal tissue. And then this is, of course, is GWAS. So in our GWAS approach, we're looking a SNP at a time, and we're protecting against all of these multiple tests. So that's all we really have. At least we think that's all we really have. But we think it might actually be fairly myopic. And again, when I said we're trying to be creative and try different approaches, we're trying to pay attention to other disciplines that are out there and see if we can't bring something else to battle here. So the compute power is amazing, what's available now. This is a graph showing the world's fastest computer from between 93 and 2018. And if you look at the rate of growth over this time period, it's 2.7 million times greater increase in compute. 2.7. You've heard of Moore's Law? It's blowing Moore's Law out of the water. It's just the advances are incredible. And in fact, in 97, the world's fastest computer was, I think it was called Big Red. It was the size of 2 tennis courts. It cost $50 million to build. To power it was the equivalent of the electricity needed to power 50 homes. That's your iPhone, right? So it was $50 million at the time. Now for $1,000, we carry it around in our pocket. That's what we've got in terms of compute power. So we're thinking, well, that's interesting. Can we use that? Well, yeah, we can, especially if we think about how we analyze the data. And so I don't know about you, but I was not paying attention at all to what was going on in computer science. And artificial intelligence has been around for a long time, since the 1950s. You know, how do we engineer computers to think like with machines and with programming? And so then we got into machine learning around the 1980s, and that's made some progress. But there was a fundamental shift when deep learning was discovered. And so being able to train computers to recognize an algorithm, and then computers find ways to recreate that pattern and make new discoveries in ways that our human brain cannot fathom. We cannot follow how they do it. But if you read the literature, do some Google searches on artificial intelligence and deep learning and how it's being developed to enhance, say, pathologists make a diagnosis. And there are examples where it's actually the computer's doing better than humans. Not that they will replace humans, but they can certainly enhance our ability to get to the answer much more quickly. So we're thinking, maybe we should take some of that and apply it to these large data sets, including GWAS. So that's what we are conspiring to do. So you might be seeing something from me on that one. OK, so what? Does anybody know where we're going with this? Why does this matter? It has to matter, right? It's not just intellectual curiosity and we're doing this for fun and we're publishing papers and getting grants. At some point, you have to say, we're doing this for a reason that's actually going to help people. So this is where I take license, and I get to just go out on the deep end. So this is not necessarily my science. This is Tom as the center director and just dreaming, OK? Is that all right? You're with me? All right, let's go for a little journey here. So translational potential. I'm spinning a little bit of a yarn here. One is this concept of risk-based prevention. Okay? I want to talk about early detection, which is a real issue for us with ovarian cancer. And then talk about what we've learned with discovery of these loci that are involved in non-coding genomes. And can we think about using that for therapy? Okay? Okay. So biggest criticism for GWAS studies is that it's not useful information clinically. Right? It's like, oh, they're a waste of time. We haven't learned anything. There's no value. And if you look at a single SNP, even though the p-value may be 10 to the minus 32nd, the risk level is 1.02. Okay? Yeah. No, you're not going to do genetic counseling for anybody on that kind of a SNP. But you can create a polygenic risk score. And anybody here worry about prostate cancer? So, and anyone know how many SNPs there are for prostate cancer susceptibility? Higher? Good guess. Anyone? It's over 200. It's a more common disease. They've got larger sample sizes, so they're getting further under the, the surface of the iceberg, right? And have identified that there's over 200. And you can really see broad differences from the low end of the spectrum to the high end. This was a paper that was published in JAMA Insights recently talking about a polygenic risk score. And for ovarian cancer, women who have 20 variants, actually just by typing 20 variants, we can identify the top 1% of women at 5% lifetime risk. So it used to be in the olden days that 10% was lifetime risk that was high enough to do some sort of intervention. But I think now there's some evidence that maybe we should be lowering the threshold. And I've heard numbers, people presenting the case at 4% lifetime risk if you can predict that. So that's with 20 SNPs. We're in the 30s. We can get higher. We can improve it. And the more that we can do that, you can lower the bar from the top 1% to maybe the top 10% to the top 15%. Because when we talk about interventions, there's risks as well as benefits. And that's part of the equation that we're trying to balance here. And that's illustrated masterfully with this figure, right? Okay, thank you. So the basic concept is if we're talking high risk, Mendelian disease, for example, you can be aggressive because you know that the risk is high enough that if we don't do something, there's a really good chance that this woman will develop ovarian cancer. If it's lower risk, do you want to just do nothing? Do you want to just wait, and if it happens, we'll cure you, right? The surgeon will take care of you. But we don't have good screening tests. So that's always kind of a bit of a gambit. So are there things that we can do that's appropriate for you? You're not at BRCA type level. So that's the concept. Does that seem sort of intuitive, right? Yeah, so we don't know where the teetering goes here, but that's the concept. So that's where you can think about BRCA mutation carriers. We know that, and that there's clinical guidelines now that say if you're a BRCA mutation carrier and you're through your risk-bearing years, you should consider prophylactic surgery, and fairly well tolerated. I think the complication rates are really, really low on this. But there's side effects. If a woman has her ovaries removed, that's a source of estrogen. Doesn't feel so good. So well, maybe take HRT. Well, HRT we know has some risks and benefits, and may be beneficial for cardiovascular, but nobody's sure about that anymore. Breast cancer, maybe your risk is elevated there. So that it's something that you have to have a conversation about, but there's some guidelines around that. Well, what if it's polygenic, so these SNPs that I've been talking about, not in a gene, but in a coding region that collectively may add up to you're at 4% or you're at 10%. You may be at the point, just based on SNPs, even without a mutation in BRCA1 and BRCA2, where, hey, let's have a conversation about having prophylactic surgery. Right? I mean, I think it's worth sharing that information. Women should know, and they should be given the opportunity to decide. For some who are going to be really afraid of this, and they've seen family members die, it's like, oh, I don't want to go. I've seen, how this plays out. I've seen that movie. I don't want to go down that path. But there are things that you can do. I mean, oral contraceptives, really effective chemopreventive agent, right? And so women who have been used OCs for at least 6 months have a decreased risk, 50%, 60%, that lasts for decades. Now, the formulations are changing over time, so it's an ongoing story. We need to continue to monitor that. And oral contraceptives have some risks as well. It's not bad advice to avoid HRT, but if you've had your ovaries removed, you're going to need some HRT. Maintaining ideal body weight, easier said than done. We're really good at this country about putting on a pound a year. Easy, right? Just slight energy imbalance, and 10 years later, it's like, where'd that come from? Yeah, I've got another 10. Close surveillance would be great, although we're fairly limited in terms of what we can do and being able to detect it earlier. But nonetheless, I'm painting a story where there's hope. And unlike I mentioned prostate cancer, 200 risks for prostate cancer, what do we do to prevent prostate cancer? We don't really have anything good for some cancers. So ovarian cancer, I think, is an exception where there are things that we know work and should be effective in lowering risk. Okay, let's talk about early detection. There is no approved screening for epithelial ovarian cancer. And unfortunately, most women are diagnosed in advanced stage, where there's, I think with surgery, chemotherapy, we can extend their life, but we don't really get to think about curing very many of these women. The survival curve goes out there tough. Stage is strongly associated with poor survival. So if we could detect at an early stage, get them to the surgeons can remove it, that's really good news. So that would be better. Prevent it completely would be great, but if you can't prevent it, detect it early and let's intervene. Do we agree on that? Yeah, surgeons agree, so thank you, Melissa. So one of the questions is, can insights from GWAS and non-coding RNA serve as biomarkers? So that's something that we have started on, in part focused on circulating non-coding RNA as biomarkers. So tumor cells are excreting all kinds of things, right? Mutations, microRNA, all kinds of things. It's in there, and they're packaged in vesicles, these microvesicles. So can you extract these from blood? You can. You have to be really good. It's difficult. There's different kits that you can use, and there's different rates of how high you centrifuge your samples to get to the right subsets. It's an area of active research. We put our toe in the water. We've tried some things. Hasn't quite worked out for us yet, but we're trying to recruit people that we know have succeeded. But these extracellular vesicles are secreted from all tissues, and they tell you what's going on in the environment, right? So what are the mutations? What about the reactive stroma? What about immune factors? Anybody who does not pretend to be at least some level of expert in immunology, you're lying. We're all immunologists now, right? How can you not be an immunologist with all the cool things that are happening in that space? Anybody going to deny being interested in immunology? I didn't think so. At least we're being honest here. This is good. Good dialogue. So, it's not clear to me these extracellular vesicles, they're sorted now by size, but we're learning now about the biology and what these mean. So I think it is possible that this can be used rather than CA-125, which is not very specific. In fact, it's interesting. Yesterday morning, I got an invitation to review a manuscript in which a group was looking at BNC2. Remember the first SNP identified as a circulating biomarker for early ovarian cancer detection. So somebody saw my slide. That's the only thing I can think of. They saw this, and they're going, yeah, I heard Tom talking about it. No, it was not me. A lot of people are thinking about it, but I think it's a very promising area that we need to spend some time in. Okay. What about therapy? Right? Let's go down that path a little bit. So, if you think about the mindset that we've had, how do we cure cancer? We have to eliminate every cancer cell in the body, right? So this is the poison chair, right, where we deliver chemotherapy based on the maximally tolerated dose. And we've got great drugs that will kill cancer cells, right? They're great. What's the problem? We kill the patient before we kill the tumor cells, right? So it's that delicate balance of how much can we do, how much pain and suffering do we inflict, and we're not doing that intentionally. The intentions are good, let's cure, but that doesn't work. Radiation, sure, but there's certainly consequences from radiation therapy. And when we look at our cure rates, despite decades, it's hard to think about really any solid tumors that we can declare victory on. Testicular cancer is one where we can now cure that. But heme malignancies, I think we've got some success stories there. But for solid tumors, we're extending lives, but we're not curing them. So, okay, maybe we should try something different. So I'm going to tell you a little story about my colleague Bob Gatenby, he's a radiation radiologist, head of the diagnostic imaging at Moffitt, and his undergraduate degree is from this place called Princeton. I don't know if you've heard of it. It's some school in the Northeast in engineering. So the guy thinks in 18-dimensional space. He's a mathematician and engineering. And he noticed that in terms of pesticide control in this country, in 1970, the U.S. Food and Drug the U.S. Department of Agriculture put regulations in place that said, look, Do not over-apply your herbicides and pesticides because it's working, but you're not killing all of these bugs, and you're cultivating for resistance. Give it up. Satisfy yourselves with 90%, 95% crop control, right? But don't try to cure all of it because we've got great pesticides and we can't use them because the pests develop resistance. And it turns out when you apply that line of thinking to human tumors, there's some similarities. And so they've done mathematical models. They've grown cells in culture. They've now done a clinical trial that shows, look, if you've got a tumor and the orange cells are susceptible to whatever drug you've been testing in the laboratory, and you throw the drug, it's going to kill those cells, which is great. And so you see this massive response. The tumor's shrinking. That's great. The problem is the bad actors that are not responsive to that drug have no competitors. So they were sort of being kept in that little environment. They were competing for resources. And then their competitors are gone. And so what you see, which is what we see in humans, that the cancer's back and it's really aggressive and we've got no effective therapy for it. So this is something that was based on, well, it got on the cover of Newsweek, where this notion of using mathematical models to understand, help inform our understanding of the biology of disease, but also to be thinking about treatment. And so that led to this concept of adaptive therapy. I like to think of it as incarceration. So rather than trying to kill these bad actors, let's just keep them behind bars. They tested that, and this was published last year in Nature Communications with men who had, gross term, castrate-resistant prostate cancer. Right? So it's the last line of treatment, trying to remove androgen, which is promoting the growth of the cells. And so there's a drug called abiraterone. And they said, you can look at the label, and the label says give it at this interval. And they said, well, maybe we're giving more than what these gentlemen need. Let's give it when they need it. Well, how do we know when they need it? Well, you could do imaging studies, or you could look at PSA, right? So they take these men. It was a single-arm study, open label, 16 men enrolled them. We're going to we're going to give you abiraterone, and we're going to monitor your PSA. And when your PSA starts to rise, we'll give you the next dose. And then we're going to remove it, and we're going to watch your PSA. And if your PSA stays low, you don't get another dose until it starts to go up again. And then we're going to give it at that point. And so what you see for these 16 men, each therapy is individualized to how their tumor is responding to the drug. So the typical life expectancy after you've reached this castrate-resistant is something like 9 months, 10 months. And then there's nothing that you can do at that point. So I think it's 3 years now. There's been one gentleman who's had progression of their disease, 3 years. So compared to what we would have expected based on men who have been treated previously, the p-value of that is 0.0000. And then they've gone back and done more mathematical modeling, and they think if we recalibrate a little bit, we can get these guys through 3 cycles of abiraterone, and they will die with prostate cancer rather than from it. And I think if you ask these guys, first of all, they're really happy because they're getting less drug, right? Less toxicity, financial as well as biological. They're handling their disease just fine. Now, if you can get over the concept of, what? You're not trying to cure my cancer? I've got this sort of Damocles hanging over my head here. But they're fine, and there are spokespeople for this approach. So this is Bob Gatenby, and he's got a smirk, but he really should be smiling because it seems to be working. We have 5 other trials that are now open where we're trying this earlier stage prostate cancer, breast cancer, cervical cancer, and one in ovarian cancer as well. So who knows? It could be the new paradigm for how we take care of patients. If you can cure. Immunotherapy, we're getting excited about that. We're curing some solid tumors that we never could before. But anyway, this gives us another tool in our quiver here. Anybody buying it? Nature Communications. What are the biomarkers? I do not know. All I know is that these are people who are working on these trials. They haven't been opened yet. I think one has scientific review committee review, but you have my email, so you can email me and I'll find out for you. Okay, so future. As a bleeding heart liberal, I believe in rehabilitation, right? Let's just not keep them locked up. Let's get these people back out on the street and contributing productively to society. So that's where I think RNA biology comes in here. And so this is my analogy of small molecule inhibitors. Gleevec is wonderful, right? That was a great story, but we've had to develop second generation, third generation, and that's working for heme malignancy. But we got these great drugs and do clinical trial, and we're getting small p-values, and we're extending their life 2 months, 6 months, a year, 2 years. Maybe some cures, but really it's not working quite like we thought. And that's because the human biology is complex. So you plug one of these, and new pathway gets activated, and the tumor cells find a way to thrive. So you could keep trying to plug the holes and put people on 15, 20, 50 different drugs at once. It's going to take us a long time to find those, especially because the therapies are often empirically based rather than using mathematical models or simulations or whatever. So I just thought simply, well, if we think about RNA biology, and a lot of this is downstream from RNA transcription, what if we just went to the source and you could turn off? Because each RNA molecule can influence thousands of downstream effectors, right? So shut down the pathway. I'm just a simple country epidemiologist. I have no idea if this made any sense at all. But thank goodness for, what did we do before the internet? I mean, I remember going to the stacks in the library and flipping through cards and finding stuff. Now, good grief, you can just hit the internet and you can find things like, wow, there's stuff in clinical trials. People think about it long before I did, but antisense and RNA interference and aptamers and microRNA, and it turns out some of these are FDA approved. So this Canamro, I won't even try to say that again. It's an antisense strategy that is for people with familial high cholesterol, and it's an injectable. It was approved by the FDA in 2013. So again, you're going to try everything that you're supposed to try, and if it doesn't work, you try this, and it works. Very cool. Now there's an FDA therapeutic against using an RNAi strategy. And this is, again, not cancer, but polyneuropathy of hereditary trans-3-mediated blah, blah, blah. I have no idea what that means. But to me, it says, yeah, we're doing it. Somebody's figured out how to make, and I think this is actually a pretty rare condition. But nonetheless, people are figuring out how to make it work. And I thought, well, what about cancer? And based on the biology, there's lots of things that one could target here. And just found this recently in Science Translational Medicine, published a few years ago by Anil Sood and colleagues, George Kalin, so pretty smart people who've really been out in the forefront in this field, where they did a nice review about can we drug the undruggable using RNA strategies. And then just recently, December of 2018, the Genetic Engineering and Biotechnology News said, here's the top 10 RNA-based biopharmas of 2018. And here they say, it wouldn't be a stretch to call 2018 the year of RNA-based treatments, such as those applying RNA interference or small interfering RNA. So it's reached the tipping point. And there are companies out there that are focused primarily on cancer and bringing it. Is it the be-all, end-all, the final solution? No. But it's nice to just get out of the paradigms that more drugs, more radiation, what's the right combination? You have to say after decades, we haven't really achieved what we want. Let's try something different. And it may be that these will be used in combination with chemotherapy and radiation. So we need to just get smart about that, be aware of the opportunities, and do what we can to keep pushing the field forward. So my conclusions. Hopefully I've convinced you. Genetics still matter. I feel like a dinosaur sometimes because it's not cool to be a geneticist. Everybody's off doing other things. They're all immunologists or whatever it might be. But I think genetics still matter. I hopefully have convinced you that genetic markers can be used to identify women at high risk, as well as for other cancers. And that can help balance the risk-benefit of the preventive interventions that we have good evidence works. We do still need to improve our early detection of ovarian cancer, and I think focus on RNA would be really important. And then, man, I'm really excited about where RNA biology can take us in terms of therapeutics, and that is something where you'll have to have another speaker come and talk about that who really knows what they're talking about. I just know enough to be dangerous. And at that point, I will thank you for listening, and I'm happy to take any questions you might have.
Who would like to start? Thank you. That was fantastic. I wanted to ask you, so we see these women who come in and, you look at their
family history. So you talk about genetics, and we see it every day. And I send them off for genetics, and I said, there's no way that you don't have something going on because your mother had it, your sister had it, your grandmother has, you know, this, and then somebody else has prostate. Can you look back at these genetic profiles for these patients and get the, I mean, Myriad Genetics has done all of our testing for how many years, right? And has to have this database of, that you could tap into, because there's just, these are the people that we should be targeting, right? I would think that these are the low-hanging fruit families that we could look at, or you could look at?
I agree completely, and I have colleagues. Alvaro Montero is one of my colleagues who started in the BRCA field. We're doing research on these variants that don't lead to a truncation of the protein. Are they functional? And it's very, very tedious to work through those. So some of these families may have a variant of unknown significance that turns out down the road, well, it actually is, and now we know the mechanism. Getting the data from Myriad has proven to be not very easy. So there's, you know, that's an issue. One of the things that this question reminded me of is when I started here in '98, they told me about a study that was done by V. Elving Anderson, where for his dissertation he did a study of like 500 consecutive breast cancer cases who were seen at the clinic, and he took detailed 3-generation pedigree, measured the risk of breast cancer in the husband side of the family, and compared them. It was one of the first studies that ever showed breast cancer clustered in families. And they said, Tom, you should follow up on these pedigrees. All of the records had been in storage in the botany building, which is no longer here, for 45, 50 years. And they said, Tom, nobody moves from Minnesota. I'll bet you can find these people. And I said, hire me and we'll do the study. And they did, and this is before HIPAA. Right? So you could call these small Minnesota towns. Are you related to so-and-so? Oh, no, that would be my cousin. You know, not my cousin, but my neighbor. You got the wrong one. Here's their phone number. Like, Minnesota, nice. It blew my mind. But, yeah, we could follow them. And when the reason why I brought this up is when you looked at the pedigrees with breast cancer and you looked earlier and earlier, we had the data. The most common cancer was? Anyone? It was stomach cancer, which was the most common cancer at the time. But, you know, stomach cancer went way down for reasons we don't know, and so it's this notion that we've got a genetic predisposition, maybe it's DNA repair. What are you exposed to? That will influence the phenotype. So I don't know what's going on with those pedigrees. There may be RNA alterations, and we're starting to identify those, or a combination of risk SNPs that push somebody over the threshold. But, you know, the issue is if there's multiple SNPs and it's segregating through families, it's not going to be one of those, you know, Mendelian-looking families. So I don't know.
Hi, yeah, you mentioned using multiple genotypes to determine like a risk score for identifying those at high risk. I was just interested in, are you also looking at environmental factors to enhance that risk score? Because it sounded like maybe it focused on the genes only in that.
Oh, that's a good question. No, we do include environment and things like nulliparity and OC use. There are things that you would want to measure, and we do have that on our studies. Now, when you talk about the Ovarian Cancer Association Consortium, there's multiple studies. People are not collecting necessarily the same thing or in the same manner. So harmonizing it has proven to be a very non-trivial task. But yes, it absolutely is measure environmental non-genetic risk factors as well as germline. Thank you for bringing that up. I should have mentioned it.
Other questions? Comments?
All right, I'll get old school. Is freckling associated with ovarian cancer risk?
I've not seen it in any studies. I don't think we've been measuring it. Certainly none of the studies that I've been involved in. But the biology is kind of interesting. It turns out that there is a fish model where they have alterations in BNC2, and they have infertility. The fish do. So there's some issues that might explain biology. Now, why freckling is related to infertility, who knows? Only the fish can tell us. But
so look in the cohorts. They've got the pigmentation data, right?
Absolutely.
Anyway, so the other question I have is about the, using genetic information to intervene for prevention, and I'm wondering if there's success stories on that, right? So, we know variants that predispose to smoking behavior and that you could modify. So, are there examples? especially in the risk window, whatever that.
That's a great question. I wish I had an answer for you. None that I'm aware of. I know Peter Konetsky, my colleague at Moffitt, is doing an intervention now on MCR1 genotype, and will that influence sun behavior? It's a risk factor, risk SNP for melanoma. We have to pay attention to the fact that the genotyping technology, if you think about, it wasn't that long ago, sequencing the first human genome took over a decade and $3 billion. We're talking about compute power. Now we can sequence the human in a couple hours for less than $1,000, and it's probably 100. So we're going to have the technology, and actually I've seen it. Patrick Soon-Shiong showed me a chip where he's sequencing his whole DNA and it's just carrying around on a chip, right? Right? So we'll have it at birth. Now, when do you use that information? You know, the concept of going out and having a DNA mutation generated at some point in your life would be moot. You would have it at birth or whenever, and then any genetic testing was completely in silico. So this is happening. You know, the technology is available. Are we as a society ready for that? I don't think so, but I'm old. So, you know, the younger people, what people post on Facebook, I'm thinking, are you kidding me? We're worried about HIPAA. And it's like, oh, no, here's a picture of this. And it's like, no, don't do that. But we have to be prepared for that and think about how do we integrate that in the healthcare system. And it would be great if it did inform behavior. You know, the problem is all of us have variants or mutations that predispose to one thing or another. And it's not a certainty. People really don't understand p-values. They don't understand probability. Risk is a difficult concept. And if you're worried about paying your Sears bill or getting braces on your kids, it's hard to be thinking about, oh, I've got a risk SNP that might increase my... So we're very well educated, and we worry about that. But communicating that to others, I think, is a real challenge. But somebody will figure it out.
Mentioning Sears makes you sound old. I had a serious question, though. The adaptive therapy, you know, in pediatric leukemia, it's been shown that adherence to medication is very important for preventing relapse. Are you worried that, you know, you got 16 very compliant men to follow up? Because now you're talking about a lifelong follow-up, which once you unleash that on the population, not everybody's going to comply.
Yeah, I think how one integrates that into care is an issue. I will say that one of my colleagues is working on a protocol now where it's a finger stick. So they finger stick and then they drop it in the mail and they send off for analysis to be able to monitor biomarkers over time. He's very interested in post-treatment, you know, detecting early recurrence of disease. So we have the technology to do that. There's companies out there that will make it. In this particular study, the men are really highly motivated. And so they are coming in, they're getting their blood drawn every 3 months, 6 months, 9 months. But it's something that we have to implement at the end of the day. You know, implementation science has become its own thing now, and that I think is a fine example of that. Yeah, good point.
Other questions or comments?
Excuse me. You brought up the dioxin slide, and for me, I just feel like we're having a hard time making that connection between all the chemicals we come, exposed to. So, do you have a feeling for first what the efforts are out there, and your sort of personal gut feeling on how we are going to make those connections? You know, because that's such, to me, it's such a big question, and there has to be a connection.
Yeah, so when I get on my pulpit and preach, it is extremely frustrating that policymakers will only make changes to regulations if there's human evidence. This is not the kind of thing that you can do in a clinical trial. Hey, we're going to expose you to dioxin and you don't. You know, it's kind of like smoking. Is smoking bad? You smoke, you not. We're not going to do that. long-term, I mean, what do you have to measure? Does it have to be cancer as an endpoint, or can we use in human systems, kind of like the data that we showed you? If you expose human cells and you see this kind of reaction, and it's kind of like the chain of causality. And epidemiologists, we've been for decades saying, you're irrelevant, you don't prove anything. But it's like, hey, a reasonable person would conclude that tobacco kills you if you look at A, B, C, and D. And eventually went, oh, yeah, there was a randomized controlled trial to prove that smoking causes cancer, but it got through their thick heads. I think we need to focus on the policy level and work with the FDA or the EPA or whoever, which is but it's not going to come from us. It's going to have to be from our colleagues in the community, right? Because when I would give talks to public groups and I would talk about risk factors for cancer, I would usually have one or two outraged person every time who says, you're not talking about the environment. You're not talking about herbicides, pesticides, all of these things that we're all exposed to. It's in our food supply. And I said, look, epidemiology is a blunt instrument. We can show an association if there's an exposed group and a non-exposed group. In this country, we're all in the exposed group, right? And so epidemiology isn't going to show it, and we're not going to do it in a clinical trial. So we're kind of between a rock and a hard place in getting the data that's needed to inform policy and bans. I mean, dioxin isn't really used. I don't think it's used anymore. But some of these things we're not finding until 20, 30 years later because we're not using the right system to generate the evidence. So I think with what we're learning about transcriptional regulation, I spent 25 of my years focusing on the 1% to 2% of the genome. And there's so much going on there. It's like we're starting over again. And when I think about gene-environment interactions, which is what we've written in all of our grants, right? We're going to test gene-environment interactions. It's not the gene. It's the regulation. That the environment is causing these genes to be transcribed, and the individual response to the dioxin or whatever it might be is what's varying between us. So it really opens up all kinds of great opportunities in terms of the kinds of studies that we do and how we think about it. That was a really long answer. I don't know if I got it all to what you were asking, but
it's a very hard question, but it's frustrating,
absolutely.
Because you feel like we're being exposed to all these things, and there's no way that epigenetically we're not modifying stuff.
Absolutely. So I agree.
No real answer.
Well, I don't know how much time you spent talking with elected officials, but wow, that's pretty sobering. In fact, a short story. When I first became Center Director 6 years ago, in the fine print, other duties as assigned, meant go to Tallahassee and talk to elected officials. And you could say the most compelling arguments with logic, and they'd say, yeah, right. So anyway, what we're going to do, and it was just like, why did I bother? So we sent postdocs to Tallahassee because they didn't care what I said or the CEO. We're not to be trusted. But when they listened to the postdocs, it's like, oh, these are still pure, and they haven't been tampered and corrupted by the machine. So they're our best ambassadors.
There was some hopeful news in that today. I don't know if Monsanto lost their case. Roundup was determined to be causative in cancer in a older couple who used Roundup since the 70s. Yeah, so I don't know how they did that, but
one of my colleagues just retired from academia to be an expert witness. I don't know who knows anything about Roundup, but there's money for you if you really get tired of doing this. Okay, don't give up on science. It's great fun.
Well, I wanted to thank Dr. Sellers for an excellent presentation.
Thank you.